Okay, in this lecture, we're going to look a little bit more into nutritional labels that we find on food and start breaking down the different components of food. In this lecture, we're specifically going to look at fats. So if we look at a nutritional label, this is something we're all familiar with. All of our foods have these labels. We have a lot of information on these labels. One component is the calories, and we've talked about what a calorie is and how much energy that food provides. Also, we find the fat content, and this is what we're going to focus on in this lecture. It's often broken down into saturated, trans, polyunsaturated, and monounsaturated fats. We're going to look at the definition of each of these and the chemical structures. Other important information that we find on these nutrient labels that we'll talk about in the next lecture are carbohydrates, proteins, and vitamins and minerals. So let's start looking at fats. First, going to give the different components of fats and oils that we're going to look at. We're going to look at saturated, unsaturated, monounsaturated, polyunsaturated. We're also going to look at what the definition of a trans fat is, good versus bad cholesterol, and hydrogenated fats. You've probably all heard about good fats, bad fats, and the definition of what's good and what's bad changes over time. A lot of the natural fats are avocados or coconut oil, are fats that are healthy. Our body needs fats. So these are fats that don't increase our cholesterol. Unfortunately, a lot of our processed foods that we all enjoy, our donuts and cookies, are labeled as these bad fats because they have a lot of these saturated and trans fats. So let's start by defining fats and oils. Fats are solids at room temperature. So something like butter, cream cheese, cream, meat fats, if you have those at room temperature, they're going to be a solid. These are mostly animal origin. Some things that are fat that are not animal origins are margarines and shortenings. Oil, by definition, are liquids at room temperatures. So if we think of our olive oil, our peanut oil, our sunflower oil, these are liquids at room temperature, and these are typically vegetable origins. Both oils and fats are triglycerides. Triglycerides are molecules that consist of fatty acids and glycerol. So let's take a look a little bit more at triglycerides. Again, triglycerides are composed of fatty acids and glycerol. The important part of the molecules for our discussion are these fatty acids. Fatty acids are long-chain hydrocarbons, so they typically contain 12 to 24 carbons. So these are much larger molecules than we've looked at throughout the semester. And then they have a carboxylic acid group at the end. This is different ways that we can write out fatty acids. This is all the same structure. This is all stearic acid. We can have the condensed formula that just tells us how many carbons, hydrogens, and oxygens that are present. We can do our line structure. With these line structures, each intersection here is a carbon. And then you see our oxygen and our hydrogen, our OH group at the end for our carboxylic acid group. And then a more detailed structure is our ball structure, where the black is the carbons, our gray are our hydrogens, and the red are the oxygens. So these are just different ways that we can display our molecule. Again, when we have a triglyceride, we have tri, meaning we have 3 fatty acids, and these are held together by the glycerol molecule. But again, it's these fatty acids that are important for us to understand the different types of fats. So fats can be classified as saturated or unsaturated. Saturated fats contain only single bonds between the carbons. An example of this is that stearic acid. There are no double bonds within this molecule. Unsaturated fats contain double bonds between some of the carbons. If you have a monounsaturated fat, that means there's one double bond. If you have a polyunsaturated fat, there are multiple double bonds. This is what the structure would look like in a line structure. So again, we have the carbons. When you have 2 lines here, that means there's a double bond between these 2 carbons. So this would be a monounsaturated molecule. This is a polyunsaturated. There are multiple double bonds. And down here we have our unsaturated fatty acid. Different naturally occurring oils and fats contain different amounts of saturated, monounsaturated, and polyunsaturated fats. Typically, we consider our saturated fats as our unhealthy fats, such as butter, fat from beef. They contain a lot of these saturated molecules. Something like olive oil contains a large amount of these monounsaturated fats, which are typically considered our healthier fats. Another term that we encounter is hydrogenated fats, or partially hydrogenated fats. Hydrogenation is a chemical process in which we take a double bond in one of our unsaturated fatty acids, react it with hydrogen to create a single bond. So this double bond now has been converted into a single bond. So the number of double bonds in a fatty acid decreases during hydrogenation. And this results in the transformation of an oil, something in a liquid, to a semi-solid fat. So this is something that we can do in a lab on a large scale to change the properties of our fats so that they can exhibit properties that we want. An example of this would be the cream in Oreos. So this is something that's not a fat that's not naturally occurring. It's been synthesized in the lab so that you get that consistency of that cream filling that is appealing both in terms of taste and consistency, but also is stable. You know, if you have it at room temperature, it's not going to melt. Another term that we often encounter is trans fats. This is a little bit more complicated when we talk about the structures, but it's important for us to address this because trans fats have gotten a lot of attention. When we're talking about trans fats, we're looking at isomers. So remember, isomers are two chemicals that have the same formula, but different structures. So if you look at these two fats, they have the same number of carbons, same number of hydrogen, same carboxylic acid group, but the chemical structure looks different. This is more of a bent molecule. This is a linear molecule. The location of these hydrogens is what controls these isomers. One is known as a cis isomer, one is a trans. So the important thing when we're discussing nutrition is that these trans isomers or these trans fats have a linear arrangement. This makes them more similar to our saturated fatty acids, which are unhealthy. Oftentimes, these trans fats are, again, created in a lab, created during the food processing. They don't occur naturally, and they've been shown to have a lot of detrimental health effects. So in summary of our fats, unsaturated fats are healthier than saturated fats. Our trans fats are found to be particularly unhealthy. Oftentimes these trans fats are produced synthetically. They're not found in the environment. The reason these are unhealthy is because they have that straight chain configuration, which are similar to our saturated fats. When we talk about cholesterol, cholesterol is associated with the fats that we eat. When our cholesterols are measured, they measure our low density and high density LDL and HDL cholesterol. Bad fats, or our LDL, are shown to increase if we have a diet high in those trans fats. And some countries have actually banned the synthesis of these trans fats and the production of these trans fats in food because of their health effects. Hydrogenated fats are customized fats in which we have modified molecules so that they exhibit the properties that we want. Margarine is an example of a hydrogenated fat. Oftentimes, a lot of the materials in cookies and candy bars contain these hydrogenated fats, which have been shown to be unhealthy. So, in summary, we have lots of different properties associated with fats. All fats are from triglycerides. They are formed from fatty acids. And the structure of those fatty acids changes the properties of these fats.